Okay, so we're going to talk today about, or in this video, about DNA's shape and how that shape compares to RNA. It's going to be brief, just the facts. Okay, so interestingly, no one knew the shape of DNA until this woman by the name of Rosalind Franklin and her lab partner, Maurice Wilkins, took this X-ray picture of it. Okay, um, and it looks like an X kind of shape, right? Well, she invited these other scientists, James Watson and Francis Crick, to her laboratory to look at this X-ray, and they took the image back. They're like, "Oh my God, I think we can piece out what DNA might look like, coolio." So they did, and they came up with that double helix model of DNA that we all know today, thanks to that X-ray. So Wilkins, Crick, and Watson all received a Nobel Prize for this. Do you notice who was missing? That's right, Rosalind Franklin got no recognition for her work with Wilkins whatsoever. Angry face, but later she was given the accolades that she deserved. So that's how we actually know what the shape of DNA is. Okay, now what does DNA stand for? Well, the D stands for deoxyribose, right? That's a sugar. And DNA is kind of named after RNA because we knew RNA first before we knew that there was this molecule called DNA. So DNA got named after RNA. So it's deoxyribose, ribose for the sugar. All right. And the N stands for nucleic and the A stands for acid. So DNA is a nucleic acid. So is RNA because the NA stands for nucleic acid. DNA is not a protein. DNA, it has all of the information for you to make proteins, but DNA is not a protein. It is a nucleic acid. Okay, it carries all of our genetic information, which we've already covered, and it's shaped like a double helix, right? That twisted ladder. So it is composed of, and I want you to remember that it is a molecule. DNA is a molecule. And it is composed of these groups called nucleotides. A nucleotide has three parts. There is a nitrogenous base, the bases, it's all about that base. So we have adenine, and adenine will always bind with thymine, and we have guanine, and guanine will always pair with cytosine. And if you ever have trouble remembering it, I always thought of it as A and T make the word at. So adenine always pairs with thymine, and guanine always pairs with cytosine. Number two, there's a sugar, that sugar group, and it's deoxyribose in DNA, and it's just called a ribose sugar in the RNA molecule. The third part is simply a phosphate group. So I'm going to give you the complicated shape, kind of looking shape of DNA right now. I'm not going to ever ask you to write it this way or draw it this way, but you can if you want to. Okay, so here we have one side of DNA. This is a nucleotide. There is a base, there is a sugar, and there is a phosphate. Okay, that's what a nucleotide is. The other side looks a little different. It is flipped upside down. It's called anti-parallel. So the DNA can lock together and then compress on itself. I didn't do that very well. And then compress on itself. It's what we call anti-parallel. Okay, like a zipper. One side is just the bonds between the nitrogenous bases are hydrogen bonds. And there are 3 bonds for one specific pairing and only 2 bonds for another pairing, which I'm not going to tell you yet because it'll give it away. So let's base pair, because you have to know how to do it. If I said that that was an adenine, what does adenine pair with? Remember the word, it makes a word. A pairs with, I hope you said T. A pairs with T. And notice that there are only 2 hydrogen bonds between them. That's right. So adenine will always pair with thymine, normally, and cytosine. So what would be on the other side of cytosine? What base will cytosine pair with? It will pair with guanine. All right. Now, this next base, what is the shape? What does it look like? It looks like thymine. And what does thymine pair with? Thymine pairs with adenine. What about the next base? What does that base shape look like? It looks like guanine. And what does guanine pair with? Guanine pairs with cytosine. And the last base, also guanine, also pairs with cytosine. All right, so that's what DNA looks like. You have to make sure that you understand the base pairing, that you can, if I give you one strand of DNA, you'll be able to give me the other strand. Okay, so let's, oh, I am forgetting some muy importante information. DNA has a 3' end, that's the 3' end, and a 5' end. Now, the other side of DNA, what do you think is at the top there? You think it's 3' or 5'? Because remember, it doesn't sit like this, it sits like that. So hopefully you said that must be the 5' end, and then the other end is the 3' end. The reason that's important is because when DNA replicates, it will only add bases to that 3' end. Only ever adding to the 3' end. Okay? All right. So let's do some simple base pairing, okay? Let's say this is a DNA strand, original DNA strand, and these are the bases. And we're going to, let's say the DNA is replicating, right? What base should your body put with that adenine? What should that adenine pair up with? I hope you said thymine. Okay, so now it gets to thymine. What base should it add? Adenine. It gets to cytosine. What base should it add? Guanine. It gets to guanine. What base should it add? Cytosine. It gets to cytosine. What base should it add? Guanine. It gets to thymine. What base should it add? It should add an adenine. And it gets to adenine. What base should it add? A thymine. That is simple DNA base pairing. Okay? And this is happening at about 50 to 100 bases a second. So this is happening really fast in your body, and sometimes mistakes get made, and we will cover that under mutations. Okay, so now we're going to compare DNA and RNA, okay? Because they're very similar, but they're not the same. So they're both NA, so what does that NA stand for? That NA stands for nucleic acids, right? They're both nucleic acids. They both contain genetic information. They're muy importante. They contain information that makes us us, that tells our body what to do. All right, DNA, there's one type of DNA. You only got one DNA in your body, in your nucleus. And there is some DNA that sits in your mitochondria, but that doesn't get replicated. That's a higher level discussion for another class. But DNA, in your nucleus, there's just one type. RNA has multiple types. So RNA has mRNA. If you're going to transcribe it to make a protein, you need mRNA. That's the molecule that reads the DNA. I mean, that's not true. That's the molecule that is created once the DNA is read. That's what I want to say. You have tRNA and rRNA, which is ribosomal RNA, and that's involved in the translation, the getting that messenger RNA info into an amino acid chain, which will then build a protein. So those are some differences. DNA has 4 bases. We just went over these 4 bases. DNA has adenine. DNA has a thymine. A DNA has the cytosine. And DNA has the guanine, right? RNA also has 4 bases, but not all the same. So, do you know which base differs in RNA? Do you? RNA has adenine, does not have thymine, it has uracil instead. RNA has cytosine, and RNA has guanine. So RNA and DNA have the same bases except for thymine. You'll never see a thymine in an RNA molecule. So if I ask you, is this an RNA or DNA molecule? You should, if there's a T, it's got to be DNA. If there are only U's, it's got to be RNA. All right, so if the original DNA strand was TCGA, what would we pair with it? If we were going to make new DNA, we'd pair an A, right? A G to a C to a G, and a T to an A. That's how basic base pairing for DNA. If it was RNA, if we had an original DNA strand, we'll take the same one of T, C, G, and A, and we needed to make some messenger RNA to make a protein, then what would we pair with T? What normally gets paired with T? Adenine. That's right. What normally pairs with C? Guanine. And RNA has guanine, so easy peasy, coolio. What pairs with G? C. Now, what pairs with, here's where, put the brakes on. What pairs with A? Some people automatically want to say T, and that's incorrect because you will notice there is no T. There's no thymine. RNA doesn't have thymine. Instead, it has the uracil. So for messenger RNA, when instead of a T, it would pair a U, have a U there for its pairing with adenine, okay? And that's basically the shape of DNA and how DNA differs from RNA. So if you have any questions, always let me know.